Good afternoon, everyone, and thank you for being here today with us in our VitroLife Group Academy studio. It is my pleasure to introduce you somebody that actually doesn't need any introduction, but again, it's my pleasure to introduce you Professor Carlos Simón, that actually is one of my mentors. And Professor Carlos Simón is going to talk today about the importance of the microbiome at the endometrial level. So, Carlos, whenever you want, the floor is yours.
Thank you. Thank you for being here. I'm going to tell you about a hot topic, which is the microbiome, the microbiota in general, but also the microbiome. What could be the relevance of the endometrial microbiome in human reproduction and the scientific basis of the actual test and even the interventional study that has been already produced. First, let me show you, this is my conflict of interest slide. I am not part of VitroLife Genomics, so I can just, whatever I say, you can take it. I am not involved anymore. I am doing my own foundation and some startups that they are getting started now. So the objectives of this very short presentation, 10 minutes, is just to inform you about the clinical implications of the human microbiome with a special focus on the endometrial microbiome and to present the concept of dysbiosis that can give us some answers in all pathologies such as endometriosis, such as recurrent implantation failure, and this will be the point of this presentation. So first, let me introduce with something that you know, which is we are not only surrounded by microbes, but microbes are living on us, helping us to do the most important functions. And we have as many cells as microbes, and if you weigh around 8 kilograms, 1 kilogram of your body body is because of the bugs that they are living on you. And preferentially, they are living in the gut and in the skin, but they are key to produce our functions. And you know, all of these figures you can know, but the question is why we talk about the microbiome now. And the reason is because the scientific advances allow us nowadays to know about the microbes that we have without need to culture them. We can use 16S ribosomal RNA, we can use next generation sequencing to identify and quantify those microbes. That's the main reason why this field has now exploded. And if you see the reference worldwide, they are increasing in all medical aspects. And the point here, if you consider this as every part of our body, the microbiota is now considered as part of our innate immune system. It's not like microbes that they are living on us. They are already in us, and they are the way that the first immunological signal will get catalyzed in order to go to the adaptive immune system. So this is the relevance. So let me just focus on the relevance of the endometrial microbiome in the endometrium, the existence of this endometrial microbiota. You cannot see that, but I'm going to just make a very brief summary. This indicates you the 3 stages in 100 years of the progress of this field. In 900, there was the dogma of that the uterus is sterile. This dogma came from a doctoral thesis that was produced in La Sorbonne. The director was Henry Tissier, and the end point of this doctoral thesis was the cervix is there as the Colossus of Rhodes, preventing that the microbes from the vagina will go to the uterus. From this moment onwards, the uterus was sterile. And it was like that for 60 years. That's it. In 60 years, start the microbiological culture. So people start to culture the endometrial fluid, the endometrial biopsies, and you can see all of these studies showing that, yeah, most of them, they were sterile, but there was some study that shows that 94 sterile, 10% sterile, 30%, something was going on here. But it was not until 2000, until just 20 years ago, that next generation sequencing shows exactly what I mentioned to you, the existence of these specific microbes living there, and those are the studies that came after that. So, and before the next generation sequencing, this is what we have. We used to take the transfer catheter, put it there, culture them, and if we have basically bacterial pathogens, we knew that it was a decrease in implantation rate, miscarriage. This is what we knew. But now we know exactly what is going on there. This was the very first study that we published 8 years ago, my God. This was Inmaculada Moreno study, in which this was a proof of concept, in which we just took only 30-something patients, and we obtained endometrial biopsy, and we analyzed the microbiome by next-generation sequencing after the cycle was done, and we divided the patients by the presence of lactobacillus dominant microbiome, more than 90%, or non-lactobacillus dominant microbiome. So we saw that the clinical differences were huge, from 60% miscarriage to 60%. Live birth from 58 to 6. So there was something, but of course the series was very short. You can see the colors here. Blue is lactobacillus, live birth. This is clinical miscarriage and no pregnancy. From this study, there was a pleiade of studies that they were confirming this. And at the end, what we know is this. It's not only a microbiome, an endometrial microbiome. There is a continuum of the microbiota in the female reproductive tract. And you have lactobacillus in the vagina, and then as you go to the uterus, the tube, the whole ecosystem is changing. So we know that this is what happens now. So what we did here, for instance, was just to have some data compiled. In addition to that, we were able to see for the first time what is the microbiome with a normal human pregnancy there. The embryo was there, and we saw what is the endometrial microbiome. In the same patient, we were allowed to study the endometrial microbiome with a previous miscarriage with euploid embryos and a pregnancy in which the baby went to term. Let me show you. This is a video that you can find in the American Journal of Obstetrics and Gynecology. Just very briefly, if we can have the sound for this.
Body can be found in the female reproductive system, where bacteria may facilitate or hamper human conception. Using 16S rRNA sequencing and functional metagenomics, we describe the human endometrial microbiota in a patient who had an 8th-week spontaneous clinical miscarriage with euploid embryos, and for the first time during a successful pregnancy in which the endometrial fluid was sampled at 4 weeks of gestation. Lactobacillus inners was a prevalent microbe found in the endometrium during early pregnancy, associating its presence with defense mechanisms and basal functions. The microbial profile prior to the spontaneous abortion had higher bacterial diversity and lower lactobacillus abundance, showing an unstable functional pattern characterized by transposisis and insertion elements. We found taxonomic and functional differences between the microbiota found in the endometrial fluid collected during an early successful pregnancy and prior to a spontaneous abortion in the same patient. Further investigation of the
we did further investigations, which is basically we went to the proof of concept to an observational study in which we demonstrate that the composition of the microbiota was associated with a reproductive outcome. In this study was an observational prospective study that was done in all of these clinics in these 3 different continents. The number, you have it here. This is the difference between the previous pilot study and this study that was published in Microbiome in 2022 with 342 patients. Here we ask all of these about 60 gynecologists just to obtain, in the previous cycle, they obtained endometrial fluid and an endometrial biopsy. Then the patient went to the IVF cycle, and once they have the results later on, we just analyze the microbiome and we saw the results. We use, of course, we update the next generation sequencing system. Here we use the 16S from Thermo. We have more hypervariable regions. So we improve the quality because it was 4 years after the first study. And here what we have is, at the end, 342 patients, and we just went to relate them with two two phenotypes: success, which is live birth, or non-success, which basically is no pregnancy, clinical miscarriage, or biochemical pregnancy. In a nutshell, basically what we found was that those patients that they have live birth, lactobacillus was the signature always there, which was in line what we saw in the proof of concept. Whereas when we have a clinical miscarriage, no pregnancy, lactobacillus went down and you have those pathogens that were present. Mainly Gardnerella was our killer on that. So with this, we found that this was a clear association in a prospective observational study. We could not intervene on that. However, there was a group, this is an independent group from Japan, that using the EMMA test, which is the name of the test that we use for the endometrial microbiome, those investigators, what they did was in patients with reproductive implantation failure, they invite those patients to have the EMMA test. So 64 of them, they refused, and 131, they did the EMMA test. This is not a proper randomized, but I mean, at least this is a prospective. Patients that choose not do nothing or just get diagnosed. So with this diagnosis, what they found is that in some of them, the results were normal. Some of them were dysbiotic, but not ready to be treated, or biomass, which basically means everything is fine. And 30 of them, the results were clearly abnormal. So what they did, talking about personalization, was to treat them with a personalized antibiotic therapy, not just high-spectrum antibiotics, but Gardnerella, metronidazole, streptococcus, ampicillin, whatever it's personalized. So they treat those patients, and they found out that 1 month after that, 75% of them, the microbe was gone. In 25%, the microbe is still there, so they go through a second one. So at the moment that the patient were ready, were cured, they went to the IVF, to the embryo transfer. These are the results that those authors reported in the first frozen embryo transfer. Having as a control those patients that they did anything, they don't know what the microbiome was there, compared to what happens with those that they have intervened. So as you see, the clinical pregnancy rate went from 12% to 42%. Live birth was 94% to 33.6%. These results are from Japan. You may think that the control is very basal, is very lower, but this is the result that they have in Japan, because they don't do chromosomal analysis of the embryos or anything else. So this is the results that they report, this improvement in the first attempt, but also in the cumulative after 2 embryo transfer, they were able to repeat that. So what I'm showing you here is from the founding of an endometrial microbiome, the founding of a signature, and then the first interventional study by an independent group using the EMMA test and treating them accordingly. And this is the concept of the dysbiosis that is helping us to understand these old pathologies. For instance, recurrent implantation failure that I mentioned to you, but there are other many that can be benefit. Chronic endometritis will need another talk, but we are not going to talk about that, but we have published also about that. But for instance, endometriosis, it was very interesting to see in translational medicine that those authors reported the presence of Fusobacterium nucleatum in 60% of endometriomas of the patients that they were investigated, and they produce an animal model, they induce endometriosis, and then they treat with antibiotics, and they were cured. This remains to be proven, of course, but what I mean is that now this is a hot topic, and of course we can just gain this knowledge to treat and improve our patients, those with specific indications. The study that I show you is recurrent implantation failure, but I think that other pathologies could be really effective. So in conclusion, I think that there is a pathogenic profile associated with reproductive failure that is evident at the taxa with the next generation sequencing, but has been confirmed by metagenomics at the species level. Lactobacillus is consistently enriched in endometrial fluid and biopsy when everything is okay. And those abnormal microbes that they are present there should be treated first. This is not going to be fixed with probiotics. If there is an issue, you have to treat the bad guys first in order to give probiotics later on. And this is just the basic part that we have been trying to understand how those microbes, they are acting on the endometrium. But I think that I am running out of time. And this, I just want you to show the faces of the people working in my lab now on this specific topic. Immaculada Moreno is the principal investigators, and we have predoctoral students, postdoctoral students, PhD students working on this topic. Thank you very much. And we are doing this in the foundation because now we are now devoted to do this work, to produce high scientific knowledge, and maybe to create a startup answering specific questions we are focused on the uterus, as you know, but we are agnostics about the results. Well, thank you very much for your attention.
Thank you. Thank you very much, Carlos. Fantastic talk, as always, real clear. Is there any question on the audience? Excuse me.
Thank you for presentation. What do you use to treat Fusobacterium?
We do not treat Fusobacterium. I just give you an example. Fusobacterium, I do not know what will be the first line of treatment, but it's not only Fusobacterium. You have to know that this is a nucleatum, so depending on the species level, you can fix the specific antibiotic therapy. Furthermore, we try to go to the next level, which is not to use antibiotics, because antibiotics is going to be the next pandemic in 50 years. But what we have now, this is what we have, the personalized treatment using personalized antibiotic therapy depending of the microbe at the species level.
Thank you, Carlos.
Oh, you go first. No.
Hi, Dr. Simón. We are seeing more interest, specifically in Canada, from OBGYNs interested in looking at the endometrial microbiome. And they're asking if we have any idea if there's an association between BV in a patient and then the presence of Gardnerella into the endometrium. And do we have any evidence of that a patient with BV would be likely to also have a dysbiotic endometrium?
You mean bacterial vaginosis? Yeah, because you always speak in acronyms. So what we have learned from this very first study, that the microbiome of the endometrium and the vagina, they are very similar, but not the same. In fact, if we can quantify, you can have a differences of 20%. In 80% of cases, we found the very same microbiome, but in 20%, you can have a VV and a normal endometrial microbiome or an endometritis. So, yeah, but if the questions goes one step forward, I know that is very popular, and this is my discussion with Carmen Rubio, but the bacterial microbiome, I know it's a proxy, but I will not treat my patients endometrial-wise with vaginal microbiome, because the vagina is an open organ and may depend on so many things in any different days that could vary and could be confused. I know that is getting now very fashionable to have the endometrial the vaginal microbiome and then to make decisions about that. I will not use as a proxy, knowing that they are related, but they are not the same.
Thank you, Carlos.
I know you're concentrating on microbiome. Are you also looking at viruses and looking at the balance of virus and bacteria?
Yes, we did. When I was at the genomics, in fact, we have already a panel of viruses. The problem is what to do with these results, because we were able to diagnose that, but if you go to a patient that want to check the microbiome and you see that she has herpes virus in the endometrium, the patient is going to be very worried. But maybe, what is the real impact of these viruses in the endometrium? We do not know. So we rather prefer not to launch this, because we cannot make any action on that. Maybe it's completely irrelevant, but yes, we have found viruses, we have found all the microorganisms, but for now, this is what we can know, and that can be and clinical impact.
More questions? So I think that I have the last one, Carlos. We are performing this kind of test, a MA test, in endometrial biopsy. But moving forward, do you think that, like in every single medical field, that we are moving into the non-invasiveness, we are going to do this kind of test in a non-invasive way?
Yes, in fact, we can do it. And I don't know why they are not doing this, because we prove in this observational prospective paper that these results were the same in the endometrial biopsy and endometrial fluid. So this is up to you to really do it in non-invasive, because to obtain endometrial fluid is so simple, and the results are published. So you can have absolutely the same. There are some extracellular microbes that they are different, but you can see the publication there. So this is up to you.
Okay, so I guess that this is the new thing, right? Okay, so thank you very much, Professor Carlos Simón, and thank you all for being here today.